Self-organization. It's a term that helps us understand how snowflake symmetry arises without a blueprint, and how ideas spread through social networks without the need for a central leader. It's the process by which order arises in a system due to the interactions of the system's individual elements, without a preset plan. Adrian Jacobo is fascinated by the physics of self-organization, and he's using it to understand something very complex: the formation of organs in our bodies.
Biology builds on relatively simple units that interact to build more complex systems, and those complex systems then build more complex systems to create amazing things that we can't reproduce with even our best engineering.
Take, for example, the human brain, a complex network made up of billions of neurons. To get here, strings of amino acids must first self-organize into proteins, proteins must self-organize into cells, and groups of brain cells must self-organize into complex information processing units.
There's no conductor, like in an orchestra, telling the cells what to do or where to go. The cells are just following these simple rules, and the collective result of these individual decisions is a very complex structure.
Jacobo is trying to parse some of that complexity, starting with an organ that, relative to the human brain, is quite simple. It's called the lateral line, and Jacobo studies it in a tiny but fascinating animal called the zebrafish. The lateral line runs the length of a zebrafish and lets the fish know what's happening around it by detecting motion in the water. Jacobo finds it a particularly useful model for study because it's a simpler analog of the cochlea of our ears, which allows us to hear by detecting vibrations in the air, and because zebrafish have a superpower: their organs can regenerate. That means Jacobo can study not only how the lateral line self-organizes during development, but also how it re-self-organizes when the organ is damaged. Key to the lateral line's function are hair cells, so named because of the tiny stereocilia that protrude out of the cell in a stair-like fashion, and which encode signals about water movement depending on how the staircase is deflected. During a fish's development, hair cells must orient themselves properly in order to encode the direction of water movements. Jacobo and his team have been able to detect just how this early orientation comes to be using the language of physics.
Physics has tools to sort of deal with some of this complexity, to do very precise predictions of how the system works.
Equations predicted that in about half the cases, lateral line hair cells will be born in the wrong position, and they'll have to flip to correct the error. And that's exactly what the researchers see happen under the microscope. Jacobo and his team hope to someday work out all the steps in how this organ forms and regenerates, a discovery they say could eventually help us learn how to promote regeneration in human tissues, to restore hearing loss and address damage to other organs.
These could have applications to help hearts regenerate and recover after heart attacks, repair, for example, kidney damage or lung damage. And I think that's something that is going to really revolutionize medicine in the next maybe 10, 20, 30 years.